Hello everyone. So this is the first chapter of the second section of the course. We are continuing to examine the cellular mechanisms that contribute to learning and memory. And in this chapter, I'm going to introduce the idea that LTP is not a singular phenomenon that onsets when stimulation occurs and simply lays down a pattern of enhanced synaptic communication. Rather, there are distinct waves of activity in LTP that can be thought of as stages or phases, where a cascade of mechanisms are employed. These stages include an initial stage wherein kinase-mediated alterations in existing proteins bring about reversible enhancements in the postsynaptic response to presynaptic stimulation. There's a second stage in which new proteins may be synthesized from pre-existing RNA. And there's a third stage that requires signaling through to the genome. The first stage of LTP encoding is known as short-term potentiation, and it lasts for about 15 to 30 minutes. The second stage is part of the late stage LTP, and it lasts for about 2 to 3 hours. And the third stage is fully late stage LTP, and it lasts 6 to 8 hours in the hippocampal slice, being limited really by how long that ex vivo tissue remains functional. Hypothetically, late LTP can last from weeks to a lifetime in a healthy intact animal. We can explore these stages and some of the underlying mechanisms using three models of LTP induction known as LTP1, LTP2, and LTP3. I have to pause and remark about what an imaginative set of names that is, LTP1, 2, and 3. Some poor unrecognized scientist really was expressing his creativity with those names. Not. Well, anyway, I modified one of the diagrams from your textbook to depict the fundamentals of LTP1, LTP2, and LTP3, and to illustrate the principles that we will talk about in this chapter. The early stage of LTP consists of the post-translational modifications of proteins that I spent much of the first 4 chapters talking about. The second stage includes new protein synthesis, but does not require genomic signaling, and the third stage requires genomic signaling. And of course, these mechanisms of LTP all have implications for our understanding of learning. There are many lines of evidence that these same mechanisms underlie various kinds of learning in intact animals and even in humans. And we'll delve into all those data in later chapters. But for now, let's examine these stages. An interesting series of studies depicted on this slide reveal once again that LTP is not necessarily stable after it is first induced. But in contrast to the previous preparations we saw, where LTP was disrupted by addition of depotentiating low-frequency stimulation, in this case, there was actually no disrupting influence. There was just a difference in the stimulation parameters. Abraham and collaborators from Mike Dragunow's lab stimulated some hippocampal preparations with standard strong tetanic stimulation. And you can see the robust LTP represented by the closed circles in the graph at the upper right of the slide. But then they stimulated additional preparations with tetanic stimulation, but in this case, they used stimulation that was not quite as intense. It's labeled on the graph as weak stimulation. But I don't want you to confuse it with weak low-frequency stimulation that's used to induce long-term depression, or LTD. This is actually weak tetanic stimulation, or maybe it should be something like tetanic stimulation that's not quite as strong as the tetanic stimulation that induced long-lasting LTP in the upper curve, but rather slightly less strong. So it only induced a transient LTP that diminished rapidly and is represented by the lower curve on the graph. But you can see how that lengthy but, well, more accurate description wouldn't quite fit on the graph, so instead they labeled it weak. In any case, you can see on the upper graph that they were able to induce a form of LTP that looked almost as strong initially, but then faded away over the course of a few hours rather than continuing for the duration of the viability of the preparation. And the lower graph on the slide extends the concept of LTP beyond the constraints of the duration of viability in an ex vivo section to LTP in the hippocampi of living animals. This lower graph shows LTP in a strongly stimulated hippocampus that was followed for a full year and was still going strong at the end of that year. So we can see that potentiation can be a short-lived phenomenon that lasts only a few hours or less in the weakly stimulated hippocampal slice, to LTP that seems permanent, or at least nearly permanent, after strong tetanic stimulation in an intact hippocampus of a living animal. So this brings up a question. How can synaptic potentiation endure over long periods of time in the face of molecular turnover? The modifications I described in the first 4 chapters all consisted of reversible changes. You can phosphorylate a receptor, for example, and that will cause it to be inserted into the membrane. It could even be stabilized there. But we know that phosphorylation is a reversible action. So why doesn't the synapse just return to its naive state after a while? Certainly it did in the case of the weak stimulation in the top graph, and certainly there should be a tendency to slip back to the naive state if all that was supporting the potentiation was the presence of some phosphorylated proteins. And let's just assume that the protein phosphorylation somehow became irreversible, so they never lost their phosphate group. Now that does not happen, but I'm kind of making up a fairy tale hippocampus here in which phosphorylation is a permanent change. It still wouldn't be sufficient to maintain a potentiated hippocampus. You see, all proteins have a half-life. They are eventually tagged and transported off to the lysosome to be degraded. If the receptors and other phosphorylated proteins were just taken off to the lysosome, the potentiated signal would be lost. And even if those proteins were replaced by newly synthesized proteins, the newly synthesized proteins would not be phosphorylated. So the synapse would not be potentiated just on the basis of eventual turnover and replacement of proteins. So you see, phosphorylation of proteins in the spine by protein kinase A, protein kinase B, protein kinase C cannot be the whole answer. These mechanisms are very efficient for initial induction of LTP, but something more must be going on in order to continue the potentiation of the synapse beyond the lifespan of the phosphorylation events, or even beyond the lifespan of the proteins themselves. So, what is the answer? Any guesses? Oh yeah, well, that one's on the slide too. So you can see that the answer is increases in gene expression and protein synthesis. And of course, the protein synthesis would have to include proteins that would support restructuring and ongoing potentiation of the synapse. Now, there is certainly evidence for this, as strong tetanic stimulation produces increases in hippocampal RNA, and memory is associated with increases in protein synthesis. However, a cascade of signaling that goes from synaptic mechanisms of phosphorylation to genomic signaling to activate transcription of RNA and translation into proteins still cannot account adequately for the continuing presence of LTP in the first hours post-potentiation. Observe the rapid decline in field potential responses to Schaefer collateral stimulation in the weakly stimulated synapse in the upper graph. And then consider that activation of a genomic cascade of transcription to new protein after an inducing stimulation like LTP takes at least a few hours. The decline in the signal strength in the weakly stimulated synapses in the upper graph appears to reflect the decay of the kinase-mediated signal. So if we had to wait for a genomic outcome, the potentiation would have to partially collapse and then recover. But it doesn't. Look at the upper set of points in that upper graph. They don't decline at all. So I'm going to propose a mechanism that looks something like the mechanism I'm showing on the next slide. The figure at the bottom of this slide shows a drawing I made to illustrate the three hypothetical stages. I haven't attached any specific time frame to it, but they do appear to occur in this sequential order. The first stage depends upon post-translational modifications, the mechanisms we've discussed so extensively across the first 4 chapters, and that are depicted as a reminder in the graphic at the upper right. The second stage is dependent upon new protein synthesis, but not on genomic signaling. And the third stage is dependent upon protein synthesis with genomic signaling. I'm going to explain that. If we apply strong, high-frequency stimulation to a hippocampal section, of course we would get LTP that should last for hours. Well, of course. And this is shown in the graph on this slide, where you can see that the control cells show enhanced EPSP amplitude recorded with an intracellular electrode. Now, what do you think would happen if we interfered with calcium influx? Yeah, of course, it is also shown on the graph. If you administer BAPTA into the postsynaptic CA1 pyramidal neuron, which you can do by micro-injecting it through the glass recording electrode, shown in the upper right, you can completely block the induction of LTP. I think I actually mentioned BAPTA before, but in case I didn't, BAPTA is a calcium chelator. It binds up calcium. So imagine if you inject it directly into the postsynaptic neuron and then apply tetanic stimulation. The NMDA receptors and voltage-gated calcium channels can open, and the metabotropic signaling can cause efflux of calcium from the endoplasmic reticulum stores. However, as soon as the calcium enters the intracellular fluid of the spine, it will be bound up by BAPTA and will not be available to activate calcium-sensitive proteins that we know are essential for induction of LTP. Similarly, if we inject PD98059, a drug that blocks kinases, then the induction of LTP by tetanic stimulation is also a big fail. So these experiments remind us that calcium and kinase activity are essential components of LTP induction, and that these signals and a subset of the targets they activate, like AMPA receptors, actually constitute the first phase of LTP. Based upon everything we learned in chapters 1 to 4, we know that interfering with them will prevent LTP induction. Now let's look at some additional manipulations. In the graphs on this slide, anisomycin or actinomycin were injected into the hippocampus, and the effects on hippocampal LTP were examined. So let's talk about anisomycin and actinomycin for a minute. You might recognize the names of these drugs because they're well-known antibiotics. So you might be asking, what do antibiotics have to do with LTP? Or what do antibiotics have to do with learning and memory? Well, to answer those questions, we need to examine the mechanism of antibiotic actions. When you have a bacterial infection, bacteria multiply at a very rapid rate. Under ideal conditions like moist, air-exposed, roughly 37 degrees Celsius conditions in your sinuses or in a break in your skin, or, well, especially in an idealized incubator in a laboratory, well, bacteria will double their population about every 20 minutes. So how do antibiotics fight that? They disrupt protein synthesis in bacteria. The multiplying bacteria need to engage in a lot of protein synthesis to form new bacteria. If the antibiotic can diminish that rate of reproduction, it may buy some time for your immune system to get a handle on the infection and fight back by eating up the bacteria. So the antibiotics are protein synthesis inhibitors, and they can actually be used in experimental preparations to see if biological functions rely on protein synthesis. Now, the graphs on this page actually summarize many experiments over the years, including experiments conducted by Krug and collaborators in 1984 and 1988, and by Huang and Kandel in 1994, and they were reported by Kelleher and colleagues in the journal Neuron in 2004. In the upper graph, you can see that these experimenters administered a single train of 100 Hz stimulation to Schaefer collateral inputs to CA1 pyramidal cells and found decremental LTP. I mean, there was an initial induction of LTP, and it decayed over the next 1 to 2 hours. The control animals that got the single train of stimulation are shown in the black line on the graph. And you can see that the pattern of response is similar to the pattern in the experiment by Abraham and collaborators in the second slide of this lecture. Now, I want you to notice that some of the preparations were treated with the protein synthesis inhibiting antibiotics, actinomycin, depicted in red, or anisomycin, depicted in blue. The lines for both of these treatments lay right on top of the line for the untreated controls. So it's clear that protein synthesis inhibition does not affect the early stage of LTP. In other words, the early, labile, and degrading stage of LTP is not dependent upon protein synthesis. And you might not be surprised about that, because we've already in chapters 1 to 4 discussed so much about the kinase activities that are responsible for the early part of LTP, for the induction of LTP. However, let's look at the bottom graph. In these experiments, the sections were treated with 4 trains of tetanic stimulation, a paradigm that's sufficient to induce long-lasting LTP in hippocampal synapses. In these experiments, both antibiotics disrupted late LTP. Let me be really clear about this. If you look at the graph, you can see that the early induction phase of LTP is intact. The protein synthesis inhibitors did not interrupt the first stage of LTP, just as in the upper graph. However, Then the potentiation was lost in comparison with the enduring potentiation that was seen in the stimulated sections that were not treated with antibiotics. But I also need you to pay attention to another important point. Do you notice that the effect of anisomycin, depicted in red, is faster than the effect of actinomycin, depicted in blue? There's a very fundamental reason for this. Well, all antibiotics are not created equal. Some antibiotics are protein synthesis inhibitors that specifically block translation from RNA to protein. Anisomycin is one of them. Other translational inhibitors include emmetine, cyclohexamide, and lots of others. But then there are other antibiotics that are protein synthesis inhibitors that actually work at the level of transcription rather than translation, blocking the transcription of DNA to RNA. This includes actinomycin. Let me repeat that. Anisomycin blocks translation of RNA to protein, and actinomycin blocks transcription of DNA to RNA. This is actually illustrated in the diagram on the lower left. So in the graph on the lower right, we can see that anisomycin disrupts LTP earlier than actinomycin does. And it does so because it interferes with protein synthesis that is not dependent upon genomic action. In other words, it interferes with translation of RNA that is already present. The later effect of actinomycin is due to disruption of genomic actions. In summary, the first stage of LTP depends upon post-translational processing of proteins that already exist in the synapse. The second stage of LTP depends on translation of RNAs that already exist. And I'll show you how this can be very readily available for the synapse. The third stage of LTP is dependent on a signal that must be conveyed to the nucleus and will then activate translation of specific sets of genes so they can provide additional new proteins to support the potentiation of the synapse. We have three broad waves of molecular changes in the cell, each of which plays an essential and sequential role in the organization of synaptic potentiation. And one more important point is that Huang and colleagues in 1996 showed that these protein synthesis independent and protein synthesis dependent properties of LTP are seen in all three major intrinsic pathways in the hippocampus: the perforant path inputs to the dentate gyrus granule cells, the mossy fiber inputs to the CA3 pyramidal cells, and the Schaefer collateral inputs to the CA1 pyramidal cells. So let's continue to explore these waves of biosynthetic activity that underlie your abilities to learn and encode memory storage in your brain. The cartoon on this slide is meant to portray that each spine is equipped with the machinery that can process local protein synthesis from mRNA at the base of the spine. It may not be the best representation because it's really just a silly cartoon and it requires some explanation. But other diagrams that are available on the internet get hung up on details of the mechanisms, so they're complex and don't clearly represent the point that I want to make on this slide. This simple cartoon actually does. What it shows is that at the base of each spine, it shows many ribosomes, consisting of the large 50S subunit and the small 30S subunit, or the large purple blob and the small purple blob, right? You can see those. The two spines on the right side of the dendrite are inactive, and so the polyribosomes are inactive. However, the spine at the left side of the diagram depicts a stimulated input in which the spine is engaged in activation of kinases and other signaling molecules, and the polyribosomes are actively engaged in translation of mRNA right there at the base of the spine, or even translocated to the head of the spine to synthesize new proteins from RNA that is constitutively available and activated or translated in an activity-dependent manner. The mRNAs that are available and are specifically translated by the activated polyribosomes include CAM kinase 2, AMPA receptors, and something called activity-regulated cytoskeleton-associated protein, or ARC. We've obviously seen CAM kinase 2 and AMPA receptors, so it's not too surprising that they are among the species of RNA that is readily available to be involved in potentiation of active synapses. But ARC is a new one, and I'm going to show you more about its function in some really exciting experiments that I'll describe in the next chapter. Anyway, the result of all this activity is that these constitutively available mRNAs are translated into essential proteins directly in the activated or potentiated spines long before any genomic cascades could begin to provide support to the potentiated synapses. For the next experiment that I want to show you, I first need to remind you that the Schaefer collaterals innervate both the stratum radiatum, or the dendritic field where the apical dendrites align, shown here and here, and the stratum oriens where the basal dendrites align, shown here and here. We saw that previously in the experiment where heterosynaptic LTD was first discovered. Remember that? Stimulation of the Schaefer collateral inputs to the stratum radiatum dendritic field produced depression, heterosynaptic depression, of the responses of the dendritic field in stratum oriens. So anyway, we can actually target the stimulation so that one dendritic field or the other can be selectively potentiated by the Schaefer collateral inputs. Bradshaw and colleagues in 2004 found that local application of the protein synthesis inhibitor emetine in the stratum radiatum disrupted the ongoing expression of LTP for Schaefer collateral stimulation targeted at the apical dendritic field in the stratum radiatum, without interfering with stimulation-induced potentiation of the basilar dendritic field when Schaefer collaterals targeting the stratum oriens were stimulated. These effects are illustrated in the two graphs on the left side of the figure. The lower left graph shows LTP in the stratum radiatum when stimulation was administered to the Schaefer collateral inputs to the stratum radiatum. The blue line shows ongoing LTP in the control slices that were given tetanic stimulation and no emetine. The red line shows the decaying LTP after just 2 to 3 hours in slices that were stimulated in the presence of emetine administered to the stratum radiatum. The upper left graph shows ongoing potentiation in the stratum oriens for control slices that receive stimulation to the Schaefer collateral inputs to the stratum oriens, either with no other treatment, shown in blue, and in sections that received emetine in the stratum radiatum. In other words, local application of the protein synthesis inhibitor emetine into the stratum radiatum produced a decay in LTP for stimulation of the stratum radiatum, but did not affect potentiation of the stratum oriens. The two graphs at the right show, I guess, kind of the inverse effect. If emetine is applied to the stratum oriens, it selectively disrupts potentiation of inputs to the stratum oriens without disrupting LTP of the stratum radiatum. Now, keep in mind that emetine is one of the antibiotics that I mentioned a few slides back, which blocks translation of mRNA to protein rather than transcription of DNA to RNA. So the results of this set of experiments on plasticity of the stratum radiatum and stratum oriens suggest that local application of a translational inhibitor of protein synthesis can block potentiation of synapses locally, restricted to the area where the synthesis inhibitor was applied. In addition, Bradshaw and colleagues showed that administration of emetine into the soma failed to block LTP in the stimulated dendritic fields. So taken all together, these data suggest that late LTP, the first phase of it, requires local translation of mRNA that does not involve signaling to the soma or nucleus. But those experiments don't conclusively tell us where the mRNA actually comes from. Of course, the time course suggests that late LTP starts too early for it to involve genomic signaling, as signaling to the genome would take at least 2 to 3 hours. And then the cell would need to transport the newly transcribed mRNA out to the dendritic field. Moreover, we already saw an experiment that showed that the translational inhibitor, anisomycin, had a detrimental impact on LTP at an earlier time than did the transcriptional inhibitor, actinomycin. If transcription of RNA was required to support the local protein synthesis, then there actually should not have been a difference between the timing of the effects of anisomycin and actinomycin. Nevertheless, Catherine Dickinson in David Wiley's lab did an ingenious experiment in which they autotomized the dendritic fields, meaning that they isolated them by cutting them away from the soma. If they isolated the apical dendritic field in the stratum radiatum before stimulating the Schaefer collateral inputs to the stratum radiatum, they still got potentiation that endured for more than 3 hours in the stimulated synapses. This demonstrates quite conclusively that the local synthesis of proteins from pre-existing mRNA supports late LTP in hippocampal neurons. Okay, so clearly the first part of late LTP depends only upon local synthesis of proteins from messenger RNA that already exists in the dendrites nearby. But I showed you evidence from the experiment comparing the effects of anisomycin and actinomycin that later stage late LTP requires genomic signaling, because actinomycin, the antibiotic that blocks transcription of DNA to RNA, blocks that later stage. So our next questions that you need to ask are, well, what signals the nucleus to start transcribing genes in support of late LTP? And how do these signals get to the nucleus? So the answer is that the signals must involve second messengers and secondary effectors, like the ones we saw in Chapter 3, along with transcription factors like CREB. So that's at least a preliminary answer to the question, what signals the nucleus to start transcribing genes in support of late LTP? It's the cascades of signaling, and we'll see more about those cascades in a minute. Then there's the question, how do these signals get there? For that, we actually have two models. In one model, which is shown in the upper diagram on this slide, potentiating activity at an individual synapse activates cascades of signaling, as we've already seen. An example of signaling in this model is that adenylate cyclase generates cyclic AMP, which frees the catalytic subunits of protein kinase A from the regulatory dimer. Then, in addition to local phosphorylating actions at the activated spine, some of the protein kinase A is transported to the nucleus, where it activates transcription factors like CREB, the cyclic AMP response binding protein. This is known as the synapse to nucleus signaling model, and in the illustration, the signals, like protein kinase A, are depicted as little green blobs heading down the dendrites to the soma and even into the nucleus. The other possibility is that the depolarization at activated spines causes depolarization at the soma, and this depolarization causes voltage-gated calcium channels to open, providing an influx of calcium that activates secondary effectors like CAM kinase 4 and MAP kinase, which also phosphorylate transcription factors like CREB. This is known as the soma to nucleus model, and it's shown in the lower diagram on this slide. In this case, the calcium influx is shown entering the soma, where it activates calcium-sensitive proteins like calmodulin, and even perhaps into the nucleus to activate proteins there. In both models, the genomic signaling must produce outcomes that will impact the activated spines. These are known as plasticity products, that is, mRNA or protein, and they're transported back to the activated spines. In our two diagrams on this slide, the plasticity products are shown as little purple blobs. We'll see more details about the specific plasticity products in the next chapter. For now, let's focus on the signals heading to the nucleus. On this slide, I've placed a diagram that I modified from your textbook to isolate two of the signaling pathways for the synapse to nucleus model. The diagram in your textbook on page 91 shows both synapse to nucleus and soma to nucleus mechanisms on the same diagram. I just deleted the soma to nucleus pathway so you can clearly and specifically see the synapse to nucleus model. Now please note that the diagram actually represents receptors out in dendritic spines and signals that would convey all the way to the nucleus along the dendrites, even though the diagram looks like the receptors are like sitting nearby on the soma. If I was making your textbook, I would have drawn that much more clearly. But I'm certain that you can visualize the G protein coupled receptor and the tyrosine kinase receptor sitting on a spine, and the arrows in this pathway extending all the way down dendrites into the nucleus. Okay, on the previous slide, I mentioned that one of the potential pathways for the synapse to nucleus model originates with activation of adenylate cyclase. Of course, we know that adenylate cyclase can be activated either by calcium influx or by signaling through G proteins associated with a metabotropic receptor. The cascade, starting with a G protein coupled receptor through cyclic AMP to protein kinase A, which is then translocated to the nucleus, is shown in the upper left part of the large diagram on this slide. Now, inside the nucleus, the protein kinase A phosphorylates CREB at serine 133, and CREB binds to the DNA and initiates a cascade of genomic signaling to eventually induce transcription of a variety of target genes, including genes that encode enzymes, structural components, receptors or ion channels, and so on. The latter stages of this process are depicted in the lower part of the large diagram, and the plasticity products that are generated could be messenger RNA or proteins that can, either one of them, that can be transported back to the activated spines. I'd also like you to note the similarity between this pathway and long-term plasticity in the Aplysia gill siphon withdrawal response. Remember how I described that protein kinase A was implicated in turning on transcription of genes that are implicated in restructuring of the axonal arbor in Aplysia? Well, in Aplysia, that function actually occurs when their protein kinase A activates a protein called Aplysia CREB, or ACREB. So, across eons of evolutionary time, a very similar cascade of signaling is retained in the invertebrate Aplysia and in mammals, including humans. Of course, if the mechanism is conserved across all these evolutionary adaptations and all the species that have derived from some primordial ancestor we have in common, you must imagine that this is a very important and essential mechanism. I also need you to note that the pathway from adenylate cyclase to CREB is not the only one that can produce activity-dependent synapse to nucleus signaling. There are many additional cascades, including signaling through tyrosine kinase receptors. Now, some people call them TRK receptors, and some people call them TRAC receptors, but they are, of course, the receptors that are activated by growth factors, and they signal by phosphorylating tyrosines through kinase activity in the intracellular domain of the receptor. These receptors are activated by growth factors, and yes, growth factors play an important role in synaptic plasticity. We'll be looking at the role of growth factors in plasticity a little bit in Chapter 6. Now, in this illustration, brain-derived neurotrophic factor, or BDNF, a growth factor that is really important in the support and differentiation of hippocampal neurons, activates the TRKB receptor. That induces phosphorylation of the second messenger RAS to then activate ERK kinase, not shown on this pathway, that phosphorylates mitogen-associated protein kinase, or MAP kinase. The activated MAP kinase then translocates to the nucleus to phosphorylate CREB at serine 133, the same serine that's phosphorylated by protein kinase A, and then the cascade of gene transcription and generation of plasticity products ensues. And this diagram represents the soma to nucleus model. Once again, I modified the diagram from the textbook to isolate the soma to nucleus pathways, and I removed the two synapse to nucleus pathways that were on the previous slide. Now, this time you don't have to visualize any distortion of the diagram, because this time the mechanism, the voltage-gated calcium channel, is actually right here in the soma. Now in this model, you can see that calcium influx through voltage-gated channels activates at least two signaling pathways that converge on phosphorylation of Krebs. One pathway is where somatic influx of calcium activates calmodulin, which then reaches and enters the nucleus. And then once inside the nucleus, it activates CAM kinase 4, which phosphorylates serine 133 on Krebs to activate genomic signaling. Now, your textbook also describes that calcium can get all the way into the nucleus and activate nuclear calmodulin that will in turn activate the nuclear CAM kinase 4. There certainly is nuclear calmodulin, so I won't argue the point with your textbook author, but I don't know the specific evidence for this slightly different version of the calcium to calmodulin to CAM kinase 4 pathway. So I'm not going to worry about that pathway, but I just wanted to bring it up because your textbook does describe it. Now then, the other pathway is where the influx of calcium activates MAP kinase, which then enters the nucleus to phosphorylate serine 133 on Krebs and turn on Krebs function. We also need to note that these pathways from adenylate cyclase to protein kinase A to Krebs, or from TRK receptors to MAP kinase to Krebs, and from voltage-gated calcium channel opening through calcium calmodulin and CAM kinase 4 or through MAP kinase to Krebs, are all essential components of late LTP. Because if you block either protein kinase A or MAP kinase, specifically at the soma, you will block late LTP, and you'll also decrease Krebs-dependent protein expression. And there's lots of additional evidence to support these models, but I'm going to focus on the soma to nucleus model for a few minutes. It seems that these soma to nucleus signaling pathways are particularly important because there's actually a need to initiate genomic signaling very rapidly. The cascade of signaling from kinase activation to phosphorylation of Krebs to increases in expression of downstream gene products that are implicated in synaptic potentiation can take a few hours. And the experimental evidence I've previously shown you clearly establishes that early LTP decays within a few hours. So we need to activate mechanisms that will support the potentiation starting as quickly as possible. If the signaling to the nucleus was entirely dependent on synapse to nucleus signaling cascades, that would introduce an even longer delay as those signals are slowly transported to the nucleus down the dendritic arbor. Therefore, some scientists have argued that those signals might not really be important in the initial stages of the genomic signaling, and that rather the soma to nucleus model is more important in activating that genomic signaling. Furthermore, antidromic stimulation of the axons of CA1 pyramidal cells, timed together with weak Schaefer collateral stimulation, actually produces long-lasting late LTP. So consider the graph on this slide. Bucalo and collaborators provided weak tetanic stimulation of Schaefer collaterals. In some preparations, they paired the weak tetanic stimulation with antidromic stimulation of the CA1 pyramidal cell axons. In other words, they provided enough stimulation of those axons so they fired action potentials. In the other preparations, they simply administered the weak stimulation of Schaefer collaterals. And of course, after these stimuli were administered, they resumed test pulses. The lower plot on the graph shows the responses of the preparations that only received the weak tetanic stimulation. And you can see once again that weak tetanic stimulation of Schaefer collaterals produces LTP that degrades over a period of minutes to hours. However, the preparations that received antidromic stimulation paired with the weak tetany of Schaefer collaterals exhibited robust ongoing LTP that lasted for many hours. Now, it's also known that the antidromic stimulation phosphorylates MAP kinase and Krebs. Of course, the relevance of this is that action potentials do not simply run down the axon. It wouldn't make sense that all the sodium that rushes in at the axon hillock all runs in one direction. Yes, it must go down the axon, but it also goes back into the soma, where we now all know that it will encounter and trigger voltage-gated calcium channels. This is the back-propagating action potential that I referenced in a previous chapter, and it's most certainly going to activate the soma-to-nucleus cascade of signaling to support the later phase of long-lasting or late LTP. So here we go once again. I have another opportunity to say that neurons that fire together wire together. But wait, there's still another important question regarding the fundamental function of both the synapse to nucleus and soma to nucleus models. So what is that question, you ask? Well, the question is this. If genes are being transcribed and perhaps translated at the soma to generate mRNA and/or proteins that are implicated in synaptic potentiation, how can these plasticity products be delivered specifically to the stimulated synapses and only to the stimulated synapses where they're going to be needed? Remember the principles of input specificity and spatiotemporal specificity that we learned way back in chapter 2? Was that really only a couple of weeks ago? Less than a couple of weeks ago? Okay, so we know that LTP is restricted to only the synapses that received the high-frequency stimulation, or in the case of weak high-frequency stimulation, the synapses that were paired somehow with strong stimulation or back-propagating action potentials. So how can plasticity products, mRNA, and proteins that are generated at the soma find just the right synapses and specifically function at those synapses to potentiate them and not at all the others? Wouldn't the plasticity products tend to go everywhere and potentiate all the synapses in the postsynaptic neuron? Does anybody have any ideas? How about this one? What if the stimulated synapses develop some kind of a synaptic marker or tag, so local transport of plasticity products would find that marker, and only the tagged synapses would be able to use those plasticity products to reinforce the synaptic modifications only at the potentiated spines? In this way, products that are generated in the soma could be transported out all over the dendritic arbor, but they're only going to be activated at those particular spines that need to be potentiated, at those particular spines that are marked to have those plasticity products targeted to them. That would work, wouldn't it? Well, indeed, it does. And it's illustrated in this diagram that I modified a bit from a paper published by Govindarajan and collaborators. I'm sure I'm butchering the name and I apologize for that. But it illustrates that stimulation of specific synapses generates potentiation tags at those synapses. These tags would then function to activate translational mechanisms to process specific forms of mRNA or to capture specific proteins as they pass by. We're going to follow up on the mechanisms of synaptic tagging further in Chapter 6. But in the meantime, I want to make certain that we're all on board with the idea that there are two overlapping waves of protein synthesis that underlie late LTP. The first is translation of pre-existing messenger RNA that is available to be used rapidly at potentiated synapses. The second is a cascade of signaling to the genome in the nucleus to activate transcriptional and translational activities on a slower timescale. The bottom line is that the basic availability of second messengers and secondary effectors at the naive synapse is not sufficient to support the continuing potentiation of the synapse after potentiating stimulation. We need local translation of pre-existing messenger RNA to support the functional and structural changes in the spine beyond the first 30 minutes or hour. Then we need genomic cascades of new synthesis of mRNA or proteins that will actually be transported out into the dendritic arbor, where they will interact with plasticity tags at the potentiated spines to supply the needed additional substrates to support the ongoing maintenance of potentiated synapses. Okay, and we're about to enter a new topic. We're about to start talking about LTP1, LTP2, and LTP3. And our time for today is nearing a close. So I think I'll pause here. And this will be continued in Chapter 5b.